Serratia marcescens, or S. marcescens, is a species of rod-shaped gram-negative bacteria and the primary pathogenic species of Serratia. Some strains of S. marcescens produce a pigment ranging from dark red to pale pink known as prodigiosin. The structure of this prodigiosin is unknown. Small growths can be mistaken for drops of blood. S. marcescens can survive and thrive in many environments, such as water, soil, starchy foods, and the digestive tracts of some animals. It is also commonly found in homes, in bathtubs, and shower walls. S. marcescens was considered to be a harmless bacterium and was used to study transmission of microorganisms. In the 1950s, S. marcescens was used to simulate biological warfare in the United States. In the Operation Sea Spray, S. marcescens was released over San Francisco Bay Area in California. After this operation, many patients at a local hospital had developed a rare, serious urinary tract infection. The number of cases of pneumonia at these local hospitals increased drastically. From the 1960s, S. marcescens has since been recognized as an opportunistic pathogen in humans. In the fictional novel Orfeo, written by Richard Powers, Serratia Marcescens had become a hazard that had caught the attention of Homeland Security because its cells were being manipulated in Peter L's, the main character, home laboratory. This brings into question whether or not an at-home lab would be able to create a dangerous pathogen.
As far as I can tell, no. I mean, you can't buy pathogens on eBay or Amazon. It just doesn't happen like this. You need a lot of paperwork, and the most dangerous the pathogen is, the more paperwork is involved, and there is no way someone can do it, just order it on a repository and not justify the use. You always have to justify why you're using it for. But people, buy and sell things legally, so maybe you could. But to my knowledge, no, it's hard to get. That's a lot easier. That you can get on eBay. If we're talking like biosafety level 1 and 2, which are the most common and the pathogens are not dangerous or lethal, then you can buy on eBay, auctions. If some places you can pick up yourself if you can, so you get fume hoods, autoclaves, PCR machines. I mean, that would not be difficult. But if we go to a biosafety level 3 and 4, then you would have trouble, because those are big equipments, they require a lot of maintenance. You would really have to know what you're doing and how to install and have a separate place. Anyways, that would be very difficult to do. I don't think anybody can have access to it. You could build it yourself if you're good enough. You would have to have a special place to have it, and you would have to know a lot about how those equipments work. And it would be very suspicious, because the maintenance of those things are very difficult. I mean, you would have to have CO2 and oxygen and liquid nitrogen being delivered to you, and that would be suspicious. And the waste, you would have to find a way to burn your waste without nobody noticing. I mean, that would be very complicated. I don't think uncertified person would have access to. But I don't know, there's handy people out there. it would be very expensive in any case. Not knowing how to do it doesn't mean you can afford how to do it. And that would be the most complicated part, I think. Well, that would be likely to happen. I mean, people who work with those high-level pathogens, they are trained for years to do it. And they have all the equipment necessary to protect themselves. And yeah, mistakes happen, but if you're in the right environment, even if you do make a mistake, you don't threaten yourself. In a home lab, that would be difficult to prevent. So yes, human error occurs, and they could be very dangerous if you don't know how to deal with them, and if you don't have the training or the equipment to protect yourself. I did glaze through an article, cheating, and I saw that they did this experiment with different salinities of water and trying to make a strain that doesn't grow in salty water to become a strain that does grow in salty water. And it apparently took four generations of gradually increasing of salt concentrations. Well, that's what they say. That's one trait. That's pretty quick, actually. I guess depending on the trait, well, I'm sure that depending on the trait, they will adapt slowly or faster. And how gradually the change happen would also change how fast they would adapt. Usually small, gradual changes, it's easier for them than harsh changes. That's for every pathogen, not this one specifically. Yes, it is. I mean, if you think that a dangerous organism would be one that has resistance to all the antibiotics known to us. Let's put it this way. I mean, we usually add resistance genes to organisms that we want to, work with or select somehow. That's one way, and we do to one or two antibiotics. We don't pick all of them and try to put all the resistance genes there. So, but yeah, we can create something resistant to antibiotics in a lab. And we've been creating superbugs in our hospitals for a long time now. You probably saw it in the news, and that's one of the bacteria that's an opportunistic one that is problematic in hospitals because it does adapt really quick, and it can grow in different hosts. Apparently it can grow in humans, it grows in plants, and so it has several hosts. So yes, we can. And we have a lot of trouble trying to control them after they happen naturally. I don't know of any studies or any lab trying to do it on purpose. I mean, dangerous organisms already exist, and we have a lot of trouble trying to find cures. I don't think any lab is trying to create more trouble. But yes, you can. You can create a dangerous organism in a lab if you know how. No, I'm not. I don't think anybody doing it yourself can create something really dangerous, and I think if they are trying and they would not know how to protect themselves well, and they would get killed before they can create a monster. As the organism becomes more dangerous, you need more protection to be able to work with it. Otherwise, it's going to kill you, and it's going to kill everybody around you. And then it will die because it doesn't have any hosts. So that's the thing with those very dangerous organisms. Once you isolate the host, they die. And without a new host, they won't live. This organism in particular can have different hosts, but I'm not scared of home labs. It's not like a meth lab that you have a recipe and you buy the chemicals and you mix one with the other and boom, you have your product. Living organisms don't work that way. It's a lot more complicated. Yeah, I'm not scared of home labs, and there's ways that we do protect ourselves, because we can buy stuff and it's just not that easy. So that's an opinion, of course. And I think that's that.